So today we're going to talk about how to use RNA Central to explore and investigate non-coding RNA sequences. So I'm Blake Sweeney. I'm the project leader. What we're going to talk about. Oh, just before we get into it today, excuse me. Here's a link to the slides where you can see you get all the slides yourself and the various links that are in it. Also, there is a Jupyter notebook that Andrew will be demonstrating later, which uses some of RNA Central's API, and it's available there as well. So I'm just going to pause for a second just to give everyone a little more time to type in tinyurl.com/RNAwebinar23. Okay, hopefully everyone got it. If not, maybe someone could put it in the chat. So what we're going to talk about today is I'm going to give you just an overview of RNA Central, what it is, what our goals are, what we want our users to be able to do. Then Carlos will be giving you a tour of the RNA Central website and a demo of some services. And then at the end, Andrew will be giving you a practical demo of using the RNA Central's API in the Jupyter notebook I mentioned. And then there'll be time for a Q&A, probably about 15 to 20 minutes. So as an overview, what is RNA Central? It's a hub of non-coding RNA sequences. Currently, it has over 34 million different sequences from over 800,000 different species. And what we do is we go to over 51 different expert databases, and we take all of their sequences and some of the metadata they provide, and we give an integrated view of all of kind of the non-coding RNA world. Instead of having to look at a bunch of different databases for non-coding RNA specific data, we give you one place to go to. So what is in RNA Central? We start with sequences and metadata about them. That's kind of the most basic fundamental building block. So RNA Central is a database of RNA sequences. But we build things on top of that. So one of the things we do is we align sequences to the genome that they're supposed to be from. Not everyone tells you where a long non-coding RNA is found, for example. And when that happens, we align it to the genome to try and find out. We also analyze all sequences with RFAM to give them a classification, which often gives you an idea of what they do and what role there is in the cell if it was previously listed as something unknown. Then we pull kind of some richer interactions, for example, richer annotations, for example, interactions from databases that do provide that, gene ontology annotations for where there's a clear function, modifications or even orthology for a few specific ones. More recently, a couple of years ago, we started computing and displaying secondary structures using R2DT, a tool we developed to display secondary structures in a standardized layout. So if you're familiar with RNA2D, hopefully the 2D structures, hopefully the ones we show will kind of look similar to what you've seen before. And the latest and greatest thing we do actually is now we're mapping to publications. So we use the metadata we have from sequences, and we basically search for mentions of that gene or RNA in all open access literature, and we extract that information and make it available, to people to see, those papers and where they were found in it. Which hopefully helps people get to the goal, which is we want our users to be able to know what function RNA has. So in the ideal world, if you had some new sequence, you could search into RNA Central, and it would tell you what it's known to do. We're not there yet because there's lots of rich data we haven't gotten yet. We don't have the entire non-coding RNA world yet, but that's the direction we're going, and that's kind of our goal. So how does it work? RNA Central, first we get the data from our expert databases. There tends to be millions of sequences. And then we import them, we assign them all what is referred to a URS, which is a unique identifier for exactly that sequence. And then we analyze it with the various tools we use, for example, RFAM to do the classification, CPAT, which detects open reading frames, R2DT to suggest a secondary structure, and then aligning to genomes. And then we publish a new version of RNA Central with that data. RNA Central data is CC0 licensed, so you can reuse it in any way you want. We do ask that you give us credit because, as academics know, citations are a big deal and are important to how you get funded. So please cite us if you use us. And what are some use cases we've seen RNA Central being used for? So sometimes people have an RNA or a few RNAs and they want to know what they are, and so they can look them up. Sometimes people like using our tools to visualize secondary structure. So R2DT is starting to become a little more popular. It was something that was originally developed for RNA Central. Since we have a large collection of sequences, it's very common to just search to see, I don't know, I have a lot of sequences, what are they? And so just using our sequence search is a very common tool. Looking around genomes to see what's in a region in terms of non-coding RNAs. And then we provide bulk data download for all of our data. And finally, everything in RNA Central can be used programmatically. So if you're a programmer like that, that's really great. We even allow downloads of our entire database, so you could run your entire own RNA Central, which is something Carlos will discuss next. And speaking of Carlos, it is now time for him to give you a tour of RNA Central. And so I think I have to stop sharing.
Okay. Thank you, Blake. I will share my screen. You are now probably seeing my screen. So it's time for a tour in our website. So the idea here is to talk about how you can access our data, how you can find a specific data on our website, and also how you can export RNA Central data. So, well, first of all, well, probably you are aware of that, but well, this is the address of our website. And once you type this address on your browser, you will see a page like this one. And then, well, there are different ways that you can access our data. We have different tools, different links, different things that you can explore to access our data. So, for example, we have a text search, we have a sequence search, a genome browser, and here on the main menu, we will also see this link called Tools, and under Tools, we will find a link for our public Postgres database, a link for the R2DT, and the R2DT is the tool used to predict the secondary X structure. I will talk about it later. We have, as Blake said, RESTful API. The FTP server, you can find the link here under download. So, I mean, there are different ways that you can use to access our data. I will talk about each of these in this presentation. I will start talking with this topic here, the text search. So when you click here in this icon that says browser sequence, what we will see, you will see a page like this one. And here, well, as Blake said, we have more than 34 million sequence. And most probably you may be interested, I don't know, in for example, some specific type of RNA or sequence from a specific organism. So somehow we will need to filter the results. And in order to do that, well, you can use facets that you can see here on the left side, and this is the easiest way to filter results. But you can also type here some text on this input, and then here you can also use logic operators such as and, or, not, and so on. We have here this link that says how to search, that if you click here, you will see a page like this one on the left side. And then here you can see, well, I'm not expecting you to read the content of this image, but I highlighted a specific part here just to give you an example. Let's say if you are interested in sequence from a specific organism, let's say from humans, and then you can use, for example, this field called taxonomy using the NCBI identifier. And let's say you are interested in sequence from a specific RNA type. So you can use, for example, RNA type and the name of the this type of RNA. And then you will see, for example, well, as I said, the easiest way is to just try to find this data here in the facets. But if you type here that you are looking for Premierna and sequence from humans, you will see that we'll get the results filtered. And now, well, here in this example, we have more than 5,000 sequences. Now you can see here a button to download some data. So this is one of the ways to export RNA Central data. I will talk about it later. But let's say we want to see the content of one specific RNA. So if you click in one of these examples, you will be redirected to what we call the sequence page. And the sequence page is everything that you can see here on this left side. Again, I'm not expecting you to read the content of this image because especially here on the sequence page, we can show lots of different things. So that depends on the sequence that you are seeing here. Different kinds of data can be shown here. So, well, some information are basic. So, for example, this first part that I highlighted here that you can see here on the right side, these are basic information we will see for all sequence. So we will see the description of the sequence. We will see an ID for this sequence, some basic information. And then here on this table, we'll see the database that have annotated this sequence. And here, just as an example, you can see that the malacards linked this microRNA with some human diseases. What more? So you can see also, for example, gene ontology annotation. So here in this case, we know that some of the data here is automatically assigned, but some are manually created. And here's the case for this specific sequence. So we know that someone created a paper or read a paper and then manually identify this specific RNA in one specific biological function, which is great. And for example, what more? You can also find on this sequence page, in some of these sequence page that we have, you will see this tab called 2D structure. And then here on this 2D structure, you will find the secondary structure for this sequence. And here we are using this Array2DT tool. Well, this Array2DT tool, it uses some standard layouts to create this this secondary structure. If you are interested in this tool, if you are curious about how it works, please take a look on this paper. It describes everything about this tool. We also have a page describing how it works. So you can also check here this help page. For some sequence, you can see here this tab called publications. And then here we have a tool called LeadScan. And here, this is our text mining tool to search for articles that mentions, for example, here we are extracting genes, IDs, and synonyms, and then we are using these genes, IDs to search for articles on EMBL-EBI. EMBL-EBI is a project hosted here by EMBL-EBI that contains millions of articles. So we are exploring this service to bring these articles, and then we are showing here these results. And as you can see, you can also filter these results using facets or by typing here some text. There are other information that you can find on a sequence page. For example, everything annotation, interactions. You can see, for example, microRNAs interacting with the long non-coded RNAs. You can see for some sequence this tab called expression, and then we are showing expression data. So there are lots of different things that you can see on a sequence page. And as I said, that depends on the sequence that you are seeing. Sorry, I missed one slide. Okay, but let's now check how you can export RNA Central data. So as I said, you can, for example, use the text search to export our data, but only if the the number of the sequence that you need is just a few thousand, let's say. So here in this example, you can download this amount of data. So under this download button, you'll see that you can download a FASTA file or a JSON file containing these results, or you can download the RNA Central IDs. But if what you need is large amounts of data, if your search produces millions of sequence, then text search is not the tool that you need to use. Then you will need to use a different tool. And for that, you can use, for example, the public database or the FTP. So let's check these tools. Here, we will see the public Postgres database. So what is this? So we have this database that you can use that contains exactly the same data that we have on our database that we use for in our production website. So of course here it requires you that you have some knowledge on SQL syntax, but if you know basic things about SQL, this is a wonderful tool. You can access this database and you can find everything about this database here on this link. You will find here what you need to do in order to connect to this database and which tables contains the main information, let's say. And you can also find here in this link, this image that, of course, you cannot see the details here on this image, but if you check here on this page, you will be able to zoom in and out and then read the content, and then you will figure out which table contains the data that you need. So I also have here one example that may be useful. So let's say you want to export all RNA sequence that we have along with dot bracket notation. So, well, you can do that using this public database. But, well, the first thing that you need to do is to try to figure out where you can find this data on this database. And then, well, you can, for example, try to find the data here on this diagram if you zoom in and out to try to find this information, or you can just connect to this database and then run some selects. Or you can also send us a message saying, listen, I would like to export this kind of data, where you can find this information, and we will help you. But then you can just, for example, run this select command to export this data. Here, I'm just putting this command here on this file called query.sql. And then you just need to run this psql command using the credentials of that Postgres database. And this will produce a file called output. And then here we will see all the sequence that we have along with dot bracket notation. As I said, this produces large amounts of data. If not wrong, this will produce, this will export more than 20 million sequence along with the secondary extraction dot bracket notation. So that's why you need to use this kind of tool. Another possibility to export large amounts of data is our FTP archive. And you can find here the link to the FTP archive. This is the help page that explains everything that you need to know about this tool. You can find at this help page what kind of information you can find in each folder. So this is another way to export large amount of data. And now it's time for demo. So I want to show you one of the services that we have. So I will open here my browser. Now you are probably seeing my browser and the RNA Central website. And I want to talk about the sequence search. So you can find the link to the sequence search here on the home page. You can also find it here under tools. But I will click here on the sequence search. And then, well, what is this tool? So this is a tool that you can use for search for similar sequence against a comprehensive collection of non-coding RNA sequence. Under the hood, this tool uses the nHammer software that is more sensitive than BLAST, but is comparable in speed. And I will click here in one of these sequence just to explain how it works. So the first thing that you may note is that when you type any nucleotide, the tool tries to find an identical match. So this is the first thing that this tool tries to show you. And then when you click on the search button, you will see that, well, we are also sending this sequence to the RNA2DT tool. And then if it produces a secondary structure, you will be able to click here on the link, and then you will be able to see the content, the secondary structure of this sequence. As I said, if you want to know more about this, about the RNA2DT, please take a look on the help page. You can find it here, help, secondary structure. This page explains everything about the RNA2DT. Let me come back to the sequence search. So as I said, we are sending this sequence to the RNA2DT, and we are also comparing this sequence with the erythrin library. And here we are using the Inferno package. And then here we will find the similar sequence. And again, you can filter results using facets. You can filter results by typing here some text. You can sort results, hide alignments, and see other details. And of course, you can also download results. When you click here, when you click on this download button, you will receive on your local machine a compressed folder. Inside this folder, we'll find a file with the metadata of this search, and another file with the FASTA file, and two different files, one in JSON format, another one in text format, where we will see the results of this search. I said, okay, here we are just submitting a single sequence, but you can submit more than one. If you, for example, if you have a FASTA file, you can upload this FASTA file by clicking here on this button. And for example, this will work if you have up to 50 queries. But if you have a FASTA file with hundreds or thousands of sequences, you can still using our tool, but then we will need to explore the API of that tool. And in order to do that, you can find this here on Help, Sequence Search API. And then here, it explains everything that you need to know in order to explore this service. You can also find here a script, a Python script, to do this kind of thing. So you don't need to create your own script. You can just copy and paste sorry, this script. And then, well, in order to run, you will need to install two packages, requests and build Python. And then you just need to run like this. You will, here in this case, you will search for similar sequence in the entire RNA Central database. Or you can specify, I want to search for similar sequence, but only for sequence from, for example, MirBase. This is also possible. So you can also check the different database available here if you want to search for just a specific sequence from a specific database. At the bottom, we'll see the Swagger page with the actions available to explore this API. But as I said, I think the easiest way is just copy and paste this script, and then you can search for similar sequence using this script. And I think that's all from my side. So now it's time from Andrew. He will show, he has another demo for you. So please, Andrew.
So hopefully you can all see that now. So I'm going to demo a couple of the other APIs that we've got at RNA Central. So this is all kind of a practical demo. I'm going to get you doing some hands-on stuff with a Jupyter notebook in Python in a bit. And what we're going to be doing is showing how we can kind of augment the analysis of the output of a differential expression experiment. And we're going to kind of go through the motions of setting this up, but we're not actually going to do it. The differential expression workflow is in our current protocols in bioinformatics paper, which is at that first DOI link there. And you can see the kind of, it's a Galaxy workflow. You can see the sort of data flow graph on the right there. I'm going to show you where to find the input data for that in our FTP archive, just because that's an interesting thing to look at. And then we're going to use a pre-prepared set of output. So I've already run this pipeline because it takes 40 minutes or so to run. And that pre-prepared outputs on our FTP. We're just going to download it and use it. So Carlos has already mentioned the FTP archive, and oops, the best place to kind of get a handle on what's in there is to just go to the help page. So you can see the URL for it there. And when you get there, you'll see there's a table that looks something like this. And it gives you the kind of the name of the thing we're looking at, a brief description. We've also got a brief description of the formats. So in this case, we're looking for genome coordinates in GFF3 format. If you wanted to go and check what the GFF format is, you could follow this link here and it will take you to that. Or you can click the link on the right-hand side of the table, and that will take you straight into the FTP site in the relevant directory. So if you click this link here, you will see a page that looks like this. You then, in this case, need to go and find the DML and Agasta GFF file, and you can download that and put it as input for your Galaxy workflow. One thing that I will point out is that this, by default, goes to the current release, which is not necessarily always what you want. So, for example, the paper specifies release 14, I think. So if you need a specific release or some sort of legacy data, you need to go and troll through the releases subfolder on the FTP and find what you need there. So that once we've got our differential expression output, we're going to then sort of augment it with some of the data that we've got at RNA Central. And we're going to do that by exploring the REST API. So this is the help page for the API. If you follow this URL, you'll get there. Excuse me. And it's sort of self-documenting, but we're going to run through it a little bit, and you can play around with this and test it in browser. So there are actually two APIs that we're going to play with today. So there's the main RNA Central one, which I just sort of alluded to, and that's got all of the information that RNA Central knows about an RNA. So RNA type, its sequence, what species it's found in, the gene names it's associated with, which database provided it, and how many publications are associated with it. That's the kind of behind the scenes what's populating the RNA page. There's another API that we're going to look at for LitScan, which gives us a kind of a window into what the literature is saying about a given RNA. So LitScan explores the Europe PMC API to look for papers mentioning non-coding RNAs, and then we pull back some of that information. So among the things that you can find in there is just any PMC ID that mentions an RNA, the titles of those papers, and some of the sentences within the papers that mention the RNA. So we're going to have a look at some of that and see if there's anything interesting that we can find. So now I would like you to switch to the switch over to the GitHub, which hopefully you'll see something that looks like this. So in here, we've got a Python notebook, a Jupyter notebook, which I've written, and you can see I fixed the spelling in about an hour ago. And the cool thing here is we can launch it interactively, and there's two ways to do that. One is Binder, which will spin up a Docker container, and you'll be able to interact with it, but it takes a little while for that to build and everything, and if we all try and do it, it will probably upset the Binder servers. The other option, which I'm going to go for today, just for the purposes of getting it done quickly, is to open it in Google Colab. So you will need to be logged into a Google account to do this, but it's quicker than waiting for Binder to load. So what you should be greeted with is something that looks like this, and you can see the notebook there, and we just click that, and with a bit of luck, it will all fire up. So what you can see is, if you're not familiar with a Jupyter notebook, this is a Jupyter notebook. It's interleaved bits of markdown that tell you what's going on and bits of code that do stuff. So the way to work with one of these is basically to start at the top and work your way to the bottom. And if you just hover over one of the code cells, so when it looks like this typewriter font, you'll see a little play button comes up. You click that. It will warn you that it wasn't authored by Google and it might do scary things. I promise it won't. So you can just click run anyway. And then in a couple of seconds, this will hopefully fire up and download the data that we need from our FTP. And there it goes. So that's all that did was just a wget to pull down the data. So the next thing that we need to do in Python, if you're not familiar, is install and load up some libraries. So I'll just set this running and then explain what's going on. So Pandas, if you're not familiar with it, is like the standard issue data frame library in Python. It's similar to data frames in R. So we import that to be able to work with tabular data. Requests allows us to actually query the API. So that's kind of a key piece of equipment. The API has a rate limit, which is 20 calls per second, I think. It's written down a bit further down. So we're going to try to respect that with what we do today. And TQDM just gives us a nice progress bar so that we can see that things are actually happening. So now that that's finished running, you see a little green tick in the top left-hand corner. We can start having a look at some of the data. So this line will just load the data. That doesn't take very long. And then we can have a look at it. So if you load this data up, and we can maybe just copy and paste this across here so that you can see what I'm talking about. Just rerun that. You'll see this is the output of DSeq2. If you've seen that before, it should look vaguely familiar. And you can see we've got 65,000 RNAs that have been identified. And an awful lot of them aren't really differentially expressed. They've got log fold changes of NaN. So we only want to look at the interesting stuff. And one of the key ways to do that is to threshold on p-value, obviously. So we're going to use a p-value threshold of 0.01. So this is very significant. The standard threshold is 0.05, but the reason I'm doing this is because we don't have that much time, and this will just cut things down a bit. So if we threshold on that, you'll see it prints out a big old table. We've got URS tax ID. So the URS is that unique RNA identifier. The tax ID is D. melanogaster. Those together specify an RNA in a specific species. And then we've got all of the other output from DSeq2. And you can scroll past this, and we should have 36 RNAs. So that's great. We've got those URS tax IDs. We can now go and try and get some data about them. And so that's what this cell is going to do. So we define a little function here. This thing just limits it to 20 calls per second. And this is the URL that we're going to query in the API. So you can just see it's just /RNA, and then RNAcentral ID gets replaced with that URS tax ID. We pull back the result as a JSON, and then we convert the whole thing into a data frame. So I'm going to set that running, and with a bit of luck, it will only take a couple of seconds to do, because I've cut down the amount of data that we're pulling. Good. So give that a second to run. Cool. And then now we can have a look at what it's pulled back. So what you can see here is the table that comes back from the API. So we have the RNA Central ID. That's just the URS tax ID. We always get back the sequence. So the sequence in RNA. We know the length of the sequence. We know this is a short description about the sequence and an even shorter description. And we have the species as a kind of human readable name, tax ID. We have a list of gene IDs, the number of publications associated with it, its type, is active. So there are some RNAs which have been effectively deleted. And in that case, this is active column would say false. And then this is the databases that provided the RNA to us. So just jump straight past that. We can have a look at how the distribution of RNA types looks. So this is just running a group by on the type and then counting. And you can see most of the stuff in there is long non-coding RNAs. but there's a few things that have only got one entry. So I'm going to pick that microRNA, which is at the top, and we're going to have a look at it a little bit more closely. So this is that microRNA, and this is just the data that came straight out of the API. So we can at this point take a gene name from here and go and search it on RNA Central or anywhere else that you want to, maybe Flybase. Or we could take the sequence and search it, or whatever we want to do. But what I'm going to do is try and find all the publications that are associated with it. We see there should be 4. So to do that, and to get the titles and PubMed IDs, we're going to use the LitScan API. So the LitScan API is a little bit different because this is actually running on EBI search. And in this case, we basically formulate a search query where all I've done, so all this Python is doing is taking this list of genes and concatenating them all together into one big, long pile of ORs. And then we pull back the title and the PubMed ID. And all the rest of this just runs it. And then we create a link to the article as well so that we can easily go and look at it. So if we run that, we get back 4 papers. So these are the 4 papers which LitScan has identified from its exploration of PubMed Europe PMC as being linked to one of the gene names for this RNA. And so at this point, you can go follow the link and go and read up on it and see what's going on. I will say that this is not quite perfect. So there are other aliases that could exist for this that are IDs, not genes. And in that case, you probably need to go to the actual URS page and find some of those and go and search them independently in Europe PubMed Central, where you can do a lot more than just pull out the title. And so hopefully that gives you a kind of a flavor for how you can do this. And if you want to play a bit more with this notebook, you can fork the repository, you can play with it in Colab, you can do whatever you like. And yeah, I think that's it. We can go and look at one of these papers, maybe. So hopefully this will work. Yeah. And so this isn't quite the right organism, but obviously they must have said something about that fly microRNA in there. Okay. And I think that's the end. So I'll switch. It's the end of the demo anyway. Switch back to the slides. And yeah, so now we can take any questions.
So the first question I can read out. Is it possible to identify novel microRNAs identified from the RNA Central database based on sequence?
Yeah, so I think I'll answer that one. Yeah, you can do that. So what I would, what I think I've seen publications do is you take your microRNA sequences and you use a sequence search. Carlos showed, I believe he showed how to use that in one of his demos. So depending on how many you have, you either upload the file or you need to write a program if you have a particularly large amount. And then you see kind of, are there any hits? Do you find that microRNA or not? And if you don't, then it's novel to RNA Central. And I believe we import all of MERBase and MERGENE-DB. It's possible that has updated recently. We haven't caught it yet. So it would be fairly novel if you didn't identify it that way.
Right. I think in continuation of that question is that I have few novel microRNAs identified. Is it possible to identify target genes using the sequence in RNA Central?
So RNA Central doesn't predict targets for you of microRNAs. So if it was completely novel, then we wouldn't have annotations for it, and therefore we wouldn't be able to predict a target for you in your genome. There are various tools to predict microRNA targets in a genome. I can't remember the list of it right now. Unfortunately, a little embarrassing, but there are certainly tools out there that do that. Yeah.
Okay, great. And I think next question was similar to that, because now the question is about long non-coding RNA. and if you can predict the target from that. So I guess
Yeah, so we don't have prediction tools built in. For some long non-coding RNAs, there are annotations available. So if you were to put in the sequence to do a sequence search and look at the sequence page, or pull from the API, you may get some target information. That's generally, I think, in humans and kind of these more well-studied organisms. I don't know if you're working in, well, okay, rats are really well-studied, but I don't think we have those annotations for rats, for example. I think they're mostly in human.
All right, so next question is, how do we retrieve information using microRNA sequence code or gene symbol?
Yeah, so there is an example of the API, which maybe Carlos can mention, that if you have a sequence, we can look it up exactly for you and get that information. And then for gene name, you can do a similar thing to find sequences that have been called that gene. Just a general warning for people and gene names. You should really know what species you're referring to, because people do reuse gene names across species. And so that may or may not be a problem for you, depending on your analysis and what you care about. So just for example, the fly microRNAs are often called, like MER120 or something like that. And that will also be used, in humans. So that may or may not be good, but it's just something to remember if you're doing a lookup by gene name, that you should also consider the species to limit it correctly.
Right, thank you. Next question is about Jupyter Notebook. So does that mean that we can use the Jupyter Notebook initially to query and download the data frame and then import that for any further analysis?
So yeah, you can use the Jupyter Notebook to do whatever you like. So if you have, so it's predicated on having the URS tax ID, that particular one that I showed. So the API that we're exploring there requires at least a URS. So if you don't know the species, you can still do something. So if you've got data so if you've got a list of RNA Central IDs, you can use that notebook to pull in everything that RNA Central knows about that list of IDs and then go do something with it. Yeah, hopefully that answers the question.
Right, so next question. Can we search starting using species or genus in the code itself?
The API, so the API that I showed isn't the LitScan API is a search API, but the other one isn't. There is an EBI search endpoint, which is what powers the text search box on the website, which you can call programmatically to do this kind of search, but I didn't show them. So right. Yes, but not with the code that I showed you, is the answer.
One question I missed is there any database specified for aptamers? Next question is, given a gene sequence that you think is regulated by a non-coding RNA, can you use this database to identify possibilities?
Yeah, no, that's a tricky one. Not directly. I think what I would start doing is, if you have the gene sequence, you can look up like an identifier for it. So if it has an ensemble gene ID, a transcript ID, or a protein name, then you could do a search. There's some search fields we allow which say this protein is regulated by an RNA, or this is a protein which interacts with an RNA, and then try to use that to find it. But yeah, that would be a little tricky. I think this is one of those tasks where you need to integrate data from a few different resources to get a shot at getting an answer.
All right. Next question is why there is no specific database for prokaryotic microRNAs?
No one has reached out to us with a database of lots of prokaryotic microRNAs, which I'm not super familiar with, to be perfectly honest. But like, we haven't rejected it, and we haven't said, oh no, you're not allowed to be part of RNA Central. I don't know of such a resource.
Next question. How could the secondary structure of a long non-coding RNA help us identify its possible target? The information provided by RNA Central could be useful in the sense that the part of the structure that is not looped could perhaps interact with some other RNA or DNA.
So the secondary structures in RNA Central are predicted by R2DT, and R2DT uses known secondary structures to make its prediction. So it's not, the current version is not finding novel secondary structures. And most of these are actually coming from RFAM, which is a database of conserved secondary structures. Long non-coding RNAs don't generally have such a thing. So we don't really predict a lot of secondary structures for long non-coding RNAs. Maybe there are some regions which look to be similar to some other thing. Maybe someone, pulled out the intron from a long non-coding RNA as well, and so we see snoRNAs in there, right? That happens. But in general, we don't, since we're not trying to predict novel secondary structures, we don't find a lot for long non-coding RNAs. For the ones that we do predict, we do have loops there that are annotated as large loops, and so maybe that's useful to you or not. Yeah.
Okay, thank you. Next question is, can I identify embryonic stem cell long non-coding RNA?
So RNA Central doesn't yet. This is something I'm very interested in getting, is having that sort of expression information. We do have some, but I'd really like to do a much broader job, a much deeper job of getting all this expression data in. So we don't really have a good way of saying, I'm interested in the expression of things in embryonic stem cells, and then kind of filtering it down to look at the long non-coding RNAs. We do have some expression information, which I think Carlos did show a bit for, but it's not at the level that I think would be that you're looking for. We could try to help you out if you reach out to us and maybe poke around a bit, but it's not quite there yet. This is, we're thinking grants right now, and I think this is one of the next main targets, is this sort of localization, specificity information, I think is really, really key. So.
Next question is, can you please consider addition of identifying target genes using microRNA sequence in RNA Central? Because there's not many tools available.
So if this is given a microRNA sequence, find all genes, I can consider it. I can see a use case for that. And yeah, that might be useful. There are a bunch of tools, but they all probably have a lot of caveats. We kind of have to sort out what the best approach is. Anyway, really, I think the people would be kind of concerned that, like, well, we can only do it for a small set of genomes, which are well studied, instead of allowing people to update. Anyway, I'm thinking how I would actually implement it. It's a good idea. And we'll look into how to do, if we can do that in a way that's actually useful to people, or at least maybe provide documentation or something that of this is a thing people want to do. Here's a good way of doing it.
So next question, does RNA Central have data on non-coding RNA expression in different human tissues or differential expression in tissues of other mammals, or organs like brain versus heart?
We do have some. So we integrate Expression Atlas, which I think Carlos showed the widget of, which does pull out expression differences, and it does show expression of some non-coding RNAs in human tissues. I will say that Expression Atlas isn't explicitly focused on non-coding RNAs. A lot of their data is long non-coding RNAs that are kind of mRNA-ish, and like they have a poly A tail, for example. Not exclusively, certainly not exclusively. But they don't focus on non-coding RNAs. There are other resources which do look for that sort of expression information. And yeah, like I said before, I think that's really important. And that's a direction I want to go. I want to add that to RNA Central, so we can kind of integrate those resources, make life easier for people.
So next question is, how can I do a sequence search with more than 50 queries?
Yeah, I think I can take this one. I presented here, I think I didn't forget to mention that, but you can search for similar sequence if you have hundreds or thousands of sequence on a FASTA file. But in order to do that, you can't use the web interface. You need to explore the API. So we have a help page. If you check on our website, there's a help link, and then on that help link, you'll see a link called Sequence Search API. And inside that link, you will find all the info that you need to explore this API. So there's also a Python script there. You can use that Python script to submit sequence from a FASTA file. You can submit as many sequences as you wish.
Yeah, just for reference, we do get, over 10,000 a month, so we can handle a fairly reasonable load. Please don't try and upload, a million sequences.
But we have some priorities, the priority is the one that we submit through web interface. So even if you submit lots with the API, well, okay, let's say it can take longer to complete, but it's not a huge problem, at least right now, in the way that we have our sequence search.
Great, thank you. And this is the last question. Do you also know an equivalent database for coding RNA of different tissues?
So if you're asking about expression of coding RNA in different tissues, I think Expression Atlas, the EBI resource, would be a good one to start with.
All right, so there's one question. This is probably the last question that we can take. Does RNA Central has microRNA expression data of disease conditions?
No. So if you look at MERBase, it does have a link to a tissue-specific expression database. I don't know if they're, I think it's small RNA, small nc expression or something like this. They have some of that. I don't know if they have disease associations as well. Oh yeah, GTEx. Thank you. Thank you. That's a good one. Yeah, no, we don't have it yet.
So thanks a lot again to Blake, Andrew, and Carlos for webinar today, and thank you all for joining in.